Oh, and Gabby, you were here yesterday, right? Yeah. Okay, so you'll do the online assignment. Yep. Cool. Okay. Hey Jay, I'm just using Zoom to record. You guys are doing the assignment that's on Canvas.
All right, thank you.
Yep, bye. Have a good day.
You too.
Okay, I've got some videos that I want to play, and none of them are that. Hopefully we're gonna be doing this a bunch of times. Hey, Pame, the instructions for you guys are on Canvas that you can locate it there. I'm just using Zoom to record today.
Okay.
Have a good day. Bye.
Bye. Yeah.
There we go. Is that the lizard? Or is it the guinea pig? The lizard has been super active today, which means he is hungry. All right. Sorry, I had the video and it's gotten like locked in with all the other things. Okay, so Christopher and Demaya, we are doing the lab together today. If you want to go ahead, I'm going to talk you through the instructions. And so if you want to flip to the second page where the data table is, you can go ahead and be there. Okay, let's slide this up here. And then share my screen. So if you guys recall, we are not allowed at school, Christopher, we are not allowed at school to like stick everybody with the lancet and, find out our own blood types. The school said no. But I did find out one of the students was like, I wonder how much that kit costs. And he found it on Amazon for like $8 or $9. If that is something you're interested in, please make sure that your parents know what's going on. I am not recommending that you do this. Right, this is my whole like disclaimer, like all those YouTubers. But if you are planning to do that, please make sure to consult your family and make sure that that's okay with them. This video that I've got on the screen, I'll lower the lights for those who are in here. This is showing you what the kit looks like when you get it, and it might be different colors based on different brands or whatever. But you see that he has this piece of paper that has circles on it, and those circles have colors in them, right? He's also holding this green object, which is, there's a second one down here. Those are the lancets. That's the part that like you stick it there and it like shoots a little blade to give you the tiniest little finger prick, right? So that a bead of blood will come out of it. And then I'm gonna fast forward this a little bit so that we can see all the other materials. Oh, he's already started using the materials. Let me go back a little bit. Select all the stuff, guy. There's like a spot on here where you can see all of the materials he has really nicely. Okay. Now his hands out of the way. Perfect. So on this card, and I've told you that we actually got to do this when I was in school, though I don't know if my teacher just had special permission or what. Is that a different school? But we did this lab in class. She had like gloves on and she like stuck us with the thing, and then we go back to the table and pull out our blood and put it onto this card. And afterward, there was this sticky clear sheet on the that you could stick on the card. You see him, right? It's not just me imagining it. Oh my gosh, the lizard's being crazy today. So that you could save that card for later. And actually, one of my friends who was in that class with me in college I happened to see that in her wallet, she would keep that card right behind her driver's license so that like if she was ever in an accident, they would immediately know what her blood type was. I was like, that is so smart. I kind of wish that I had done that too. My results were inconclusive, so it probably wouldn't have even been helpful. But on the card that he has in the video here, you can see that each of those circles has a color on it. What he's starting to do in the video, we're not going to watch this whole video because then I can't post this recording. But we'll fast forward and take a look at the things that he's doing. The circles have different colors in them, and he's activating those right now. And we are going to see that in the lab that we do. We're going to have different blood type samples. This is fake blood, simulated blood. And then we're going to have the stuff that is currently dried up on his card. These have antibodies on them. They're proteins that recognize the different antigens. Okay, so then he goes and pricks his finger, and he actually ends up being too dehydrated when he does this. He's cleaning off. He used the palm side of his middle finger. And just FYI, your middle finger, pointer finger, and thumbs are usually the least likely to be good for this sort of thing. So you're better off using your ring or pinky finger. Simply because we don't use them as much, right? These are our 3 pincher grasps for picking things up and touching things. Okay, so as he goes through, he uses his middle finger first, and it's like, actually, I'm too dehydrated, so blood won't come out, and so now he has to prick himself again. And that's why there's the extra lancet in there. And so he goes, and then I'm like afraid to play it because then my recording won't upload to YouTube. But then he takes these 4 little white strips. We are going to use toothpicks to do this job. But he like presses one against the blood coming out of his finger and then rubs that on these circles here. Okay, so we can see that he's already smeared one of them on. And then he has the next of these little sticks, and he does the same thing on the next one, and new stick on the next one, and new stick on the next one. Why do we need the different sticks? Why can't we just use the same one each time? What's it called? If we were to do that, we'd be like putting, there we go, cross-contamination. We'd be getting the stuff from this first one on the second one, in which case we don't know, we don't have a controlled experiment anymore. It's not telling us whether it's A or B. It could just be both of them. It might just be one of them, but we wouldn't know. So that's all we need from his little video there. I might play it for you guys later as we move forward. Yeah. Yeah, copyright issues. Yeah, if I'm recording a recorded thing, it's come back before because I wasn't sure if something would play. And then like I get an email from YouTube saying, hey, we've removed your video due to copyright issues. I was like, oh my gosh, I'm going to be arrested. Let me find the my other screen. And so now we're going to start into the lab. Okay, people who are in here, you can come up, get a pair of gloves and one of these kits, and then head back to your seats. We got small, medium, large, and extra large. It's your choice which one you want to use. Nope, just any of them. Because they're all different sizes. So are those too small? If they are, then you'll need to throw them away. It's not a big deal. I would say you need at least large, if not extra large. So to organize yourself for the lab, just put all of these little trays in a row. if you hold them up to the light, you can see that it says A, B, and RH, and you want that to be in the same direction for you all the way across. So I've got all these lined up. It doesn't matter if there's like some stain of the previous lab on there. It's been wiped off enough that it shouldn't be an issue. Okay, and then take the blood samples out of the bag. Again, this is fake or simulated blood. And let's put those in order behind each of these plates. We want sample 1 first, 2, 3, and 4. So for those of you who are going to be needing this later, I've just got everything organized, separated out like this. You can get the serum out of the bags. And you'll see on the serum that there's a blue one, there's yellow one, there's a clear one, and they each say a different anti-something. So the blue one says anti-A serum. The yellow one says anti-B serum. And the white or clear one says anti-D serum. All of these have the antibodies for the blood cells. Okay, so remember that on our blood cells, we have the antigens. Those are the little proteins that are present or not present on your red blood cells. So there's the antigen for A, there's the antigen for B. Some people have one or the other. Some people have both, which would give them type AB blood. Some people don't have any of those, and so they would have type O blood. And then we also have, remember the RH factor, that's what goes with anti-D serum. And that's another antigen entirely. And if that is on your red blood cells, then you have positive blood. If it's not on your red blood cells, you have negative blood. So now with the serum, these have the antibodies. Someone with type A blood has anti-B antibodies. This one, they would have anti-B antibodies in their cells. Someone who has type AB blood doesn't have either of them. Someone who has type O blood has both of them, okay? So the tricky part here, and this is what I struggled with when I was a student learning about this, is that when we put these different serum on the plates, if there is clumping in one of them, then they have that antigen. And it's like, wait, I thought there wasn't supposed to be clumping. And the answer to that is that yes, in your body, there would not be clumping. If you have type A, your body wouldn't have antibodies against the A antigen, right? Antibodies are proteins floating around your blood saying, hey, this thing doesn't belong. If we see it, we're going to attack it, right? And have that agglutination. But on these plates here, in order to identify that something has the antigen, we need the antibodies for that antigen. So if someone has type A blood, I'm going to put this screen on the board real quick. You can refer back to it if you'd like. Looking here for people in the room. Okay, so when we are doing the blood typing test, when we see this top one, for someone who is O positive, they are not going to have any clumping for the anti-A or anti-B antibodies because their blood won't have those antigens. They won't be there to be recognized. If they have positive blood, then there will be clumping with the anti-D, okay? For A, the two types of A blood jumping down here, there will be clumping with the anti-A serum, nothing with the anti-B serum. If they've got positive blood, there will be clumping for the anti-D. If they're negative, no clumping, okay? So the big takeaway to the phrase to use to remember is that when doing blood typing, if there is clumping, they have that antigen. Okay, so going back to my screen over here. So we're going to start off, take sample 1, and then in your first plate, put a drop of that in each of the wells on the plate. be sure to like hold this far enough away that the drops actually fall to the plate. We don't want it to be too close right up there when we do any of this, because sometimes if like we're going to use the next serum, if we're too close to it, we might end up squeezing some of the liquid back into the tube, and then it ruins that solution basically forever. And these are brand new. Yesterday's people were the first ones to ever use these, and so it'd be nice if these could last a while. But if you take a look, I don't know if you guys can see on here, there's like red, somebody messed that up. It looks like it didn't get all the way into the bottle, so that's good. But hopefully we can prevent that from getting worse. You can see a little bit solidified in there. So I guess we'll find out if this particular bottle still works. Okay, if you recall, when you looked on here, it says A next to the top of the plates, B next to the bottom of the, of the wells, and then it says RH to the one on the side. Okay, you can grab one of your blank ones and hold it up to the light if you aren't knowing what I'm talking about. You see A, B, and RH. Okay, so we only want to put one of the serum in any of the wells. So I've got A to start off. We're going to put a drop of that into the A well only. We'll put one drop of the B into the B well only. Ha, B well. Yes, be well. I feel like that's like something they say to you at church, right? Be well on your way out the door. And then the anti-D serum will go into the RH well only. Again, we only want one serum in any of the wells so that we have a controlled experiment, that we're only testing for one antigen in each of the wells. Then you're going to take your toothpicks. And again, we don't want cross-contamination, so we want a new toothpick for every well. We start stirring that up. I'm going to tap that to this, like lean that over to the side so that I can use it again if I want to stir some more. Stir B. And then stir the RH. I'm going to zoom in now so that we can see this really well on here. Immediately, what do you see happening in any of the wells? Whichever one you want to talk about first. Did anything happen when you started stirring it? What happened? Or in which one? Yes. In the A well, it looks like it didn't mix properly. That's a good description. It's really easy to see. Oh, sorry, people in the room. I don't have this so that you can see my screen anymore. I apologize. Let me slide this up. Sorry. Whoops. I don't know what that just did. I need to take a picture. Oh, I thought it stopped recording. I was going to be so upset. Where's my resume? Okay. I know that this doesn't look particularly great for the people in the room looking at it on the board. It does seem to be showing up okay on my computer screen. But when you look at A, when we stir it around, it looks kind of like oil and water, how they separate from each other when they're together, right? Okay, so that is one way that it might look like clumping. What else had funny stuff going on? Yeah, the RH group with the anti-D over on the side, it became gelatinous, right? I love that word. So many syllables, right? But it's like if you took a fork across Jell-O and it's really chunky. That is another way that would be considered clumping. Did anything happen in B? Yeah, it looks like it just kind of thinned out, right? Nothing particular to note. Okay, so with that, when we get into our data table, right? When we get into the data table, we have, for sample 1, anti-A, was there clumping or no clumping? when we look back at how it was? Did we call that clumping or not clumping? Clumping. And so that's what you're going to write in this box here is clumping. For anti-B, was there clumping or no clumping? No clumping. And then in the RH, there was clumping. And so at the bottom there, it asks, based on that information, which blood type do they have? There's clumping in A, so we recognize the A antigen. No clumping in B, so there's no B antigen. And there's clumping in the RH, so there is the RH, A positive. That will be an A positive blood type. For the sake of my recording, people who are doing this in class, you will continue doing the lab. Same process, put the samples of blood in each of the wells, and then put the serum, one serum in each of the wells, and test it out, and then record. When you're done, take your plates, wash them off in the sink, dry them, just pat them dry, and then you'll see that there's a drying rack area of them on the side of the room by the windows. For people at home, I will continue doing the samples across here so that you can see what the results were. But otherwise, the people who are doing this in the room can continue doing that on your own and at your own pace. And then there's analysis questions at the end. Everyone needs to do those. I'm not collecting them back from you, but you need to do them because they will be part of your quiz tomorrow. Remember that you need a new toothpick each time in order to prevent cross-contamination, but every toothpick has two sides, so maybe we could utilize both sides when going from one sample to the next so that we aren't using so many materials. I seem to be low on toothpicks. Obviously, I'm not desperate for them, but it's nice to not have to make a second trip to the store for just toothpicks. Okay, so I'm gonna, for the people who can't come in to do the lab in person, I'm going to start setting up the other samples. Okay, I'm gonna put the blood in for sample 2. Gonna put the blood in for sample 3. You can give them a shake if you want to, just to make sure that things haven't like dropped out of solution. And then the blood for sample 4. I'm speaking out loud simply to make sure you know what I'm doing. I'm now putting the anti-A serum into each of the A wells. I'll then put the anti-B serum into each of the B wells. And the anti-D serum into the RH wells. I don't know why they call it anti-D instead of just calling it anti-RH. Do not know. Okay, so now that you've seen how I've made all those, I'm going to zoom in on number 2. We got A. Got B. I forgot to mention to the people in the room, there's one other way that it can look like clumping, because all of these are going to be different blood types, either A, B, AB, or O. And it's that if you look really closely, it looks like little granules, little like rice seeds, little tiny, 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 have separated out too. That also counts as clumping. Did you see that in the second one? Okay. And then for RH, you can see that big old chunk there. Okay, so that is the info for sample 2. Going up on sample 3. There's for A. Ooh, I'm about to stick my sweater right into that sample. Here is for B. Here's for RH. That seems to not really be doing anything. Okay. Real close. That actually helped. Okay. And then for sample 4, and then that's all of our samples. Use that information to help you fill out the other blood types. And then don't forget to finish your analysis questions. This sort of information will be on your quiz. See you later.